amazing to know. Just imagine if we can do this in human beings. We can generate energy anywhere, and it will be wonderful if we can do that. That is a real magic. Understanding is not enough. We have to really take science so far beyond our expectations that we can make completely unimaginable magical things possible. And that is when I think is the true use of understanding this concept. So don't give up. Go over the concept one more time and see if you can really understand the secret, how plants do this, and is there any way we can really make it happen in in a maybe in a little test tube, and then in a little mice, and then in a small baby, or or a model organism. And we can be millionaires, trillionaires, no gazillionaires, definitely. Well, coming back to our concept from dreams, um, light reaction uses solar power to generate ATP and NADPH. Yes, that is what happened, right? And now let's see how the energy from ATP and NADPH now will be used by plants to make their food. Cyclic electron flow. The thing is, in cyclic electron flow, which also occurs in the thylakoid membranes of the photosynthetic bacteria. They have only photosystem 1, and the electrons coming off of photosystem 1, which is P700 chlorophyll A molecules, it will simply return to it. It does not produce any NADPH because it is not given to or accepted by NADP plus. So only they're making only ATPs and living off of them. They're only making ATP. So cyclic electron flow generates a lot of ATP, and these ATPs only are used for making their glucoses in the Calvin cycle. So they're not living off of this ATP for their energy requirements. They're using ATP made by cyclic electron flow to make food in the Calvin cycle stage. Okay. So this is all the notes for your chemiosmosis part that takes place during the light reactions of chloroplast. Little more notes, detailed notes, if at all you want another version of it. I added a few more points to make it much easier. And these are some of the summary points. Electrons flow from starting from water, they end up with NADPH. And remember, photosynthesis, it is light driven electron flow, which will give energy to chloroplast to regenerate ATPs. Same process, ATP synthase, chemiosmosis. And in this light reaction stage of photosynthesis, light is converted to chemical energy, which is in ATP and NADPH for now. Light reactions do not make sugars. Calvin cycle makes sugars. During this process, water was used up, light energy was used, oxygen is released, energy products ATP and NADPH are made, but the final purpose of making sugars is not finished yet. I bet by now your minds are going crazy, and everything got mixed up. But guys, don't give up. Energy coming from sun will pass through pigment molecules to special pair of chlorophyll molecules in photosystem 2, which is P680, and then electron acceptor, primary electron acceptor, then electron transport chain 1. And remember, we are talking about energy here, so it is converted into proton motive force and will make some ATPs, but energy process does not stop there, because energy coming from sun will also energize the P700 pair of chlorophyll molecules in photosystem 2 and photosystem 1 and electron transport chain 2, and finally energized electrons or energy through energized electrons is given to NADPH, which will be finally given to glucose through Calvin cycle. So the gray part is in Calvin cycle. Coming to electrons, they start their journey from P680, which is photosystem 2, electron transport chain 1, P700, electron transport chain 2, and NADPH till the light reactions. But they do not stop there. NADPHs are, remember, they're only electron carriers. So what they do is they will finally give those energized electrons to glucose through Calvin cycle. So let's see that. Calvin cycle in short is CC. Guys, try to differentiate between Calvin cycle and citric acid cycle. Citric acid cycle takes place in cellular respiration, right? Calvin cycle is CC, which is in photosynthesis. So CAC, citric acid cycle in cellular respiration. CC is Calvin cycle, which is second stage of photosynthesis. Okay, so let's see how plants use a Calvin cycle to make sugars. Calvin cycle has 3 phases. The 3 phases of Calvin cycle are carbon fixation, reduction, regeneration of carbon dioxide acceptor by RUBP sugar. Carbon dioxide is added to sugar called RUBP. And who is going to add this carbon dioxide to RUBP? It is an enzyme called rubisco. So learn what rubisco stands for, learn what RUBP stands for. 3 phases of Calvin cycle: carbon fixation, reduction of G3P, reduction is adding energized electrons to G3P sugar, and regenerate sugar called RUBP. So let's see. Is most abundant protein in the biosphere, entire biosphere, is rubisco. Rubisco, which is an enzyme complex. What it does is it will add carbon dioxide molecule to RUBP sugar, which contains 5 carbons. So carbon coming from carbon dioxide will make it 6 carbons. As soon as carbon dioxide is added to RUBP sugar, it is broken down into 3 phosphoglycerates, 3PG, and this 3 phosphoglycerate, it will use up ATPs and will become 1,3-bis phosphoglycerate. This is just the phosphorylation step, and then this 1,3-bis phosphoglycerate is going to break down accept electrons from NADPH. Accepting is gaining, and it is reduction. So reduction of 1,3-bis phosphoglycerate to glyceraldehyde 3-phosphate, which is also called G3P. You should know what G3P stands for, guys. It is very important. Coming to the third stage of Calvin cycle, which is regeneration of carbon dioxide acceptor, which is RUBP sugar. How RUBP is regenerated. So many Calvin cycles are taking place like this. So let's take into consideration 5 G3Ps coming from 5 Calvin cycles. 5 G3Ps contain 15 carbon molecules, right? Carbon atoms in the molecule. 1 G3P has 3 carbons. Then 5 G3Ps would have 5 times 3, 15 carbons. So these 5G3Ps, they're broken and rearranged in order to regenerate 3 RUBPs. Again, each G3P is 3 carbons. There are total of 5 G3Ps. So number of carbons will be 5 times 3, 15. These 5 G3P molecules are broken and rearranged to make 3 RUBP sugars. So that is how RUBPs are regenerated. And these RUBPs are going to bump into rubisco. Rubisco will add carbon dioxide to it, adding a carbon. Remember, RUBP has only 5 carbons, right? So carbon dioxide adds 1 carbon, and then this molecule RUBP plus carbon dioxide, which is fixation of carbon fixation first step, it is going to go through a step wherein it will use the ATPs coming from light reactions and take electrons, high energized electrons from NADPH, again NADPH coming from light reactions, and then it will make G3P through reduction phase. G3Ps will be used for regenerating RUBP, but out of 6 G3Ps from 6 Calvin cycles, 5 will be used for regenerating RUBP, but 1 will be coming out and 1 from another Calvin cycle will come out, and 2 G3Ps together will make glucose. So what is the name of the sugar that comes out of Calvin cycle? It is G3P. What are the inputs of Calvin cycle? Carbon dioxide is used. ATPs coming from light reactions are used. NADPHs, which are outputs of light reactions, are used. And RUBP, which is already there in the stroma, is used. And G3Ps are made. Remember, G3Ps are made. And ADP and inorganic phosphate are released. NADP plus and inorganic phosphate are released. And this process goes on and on and on, and lots of glucoses are made, or G3Ps can be used for making whatever the plant needs. They make their own antioxidants, they make their own vitamins. NADPH, they have to make it on their own, they cannot buy it from Publix, right? They have to make their own ADPs, they have to make their own lipids, they have to make everything. But if they need phosphate, they have to absorb it from soil. They have to absorb all the minerals from soil. But the organic molecules, they make it. They make their own proteins, they make their own carbohydrates, they make their own DNA, they make their own lipids, everything. They need organic molecules, they make it out of G3P, which is the product of Calvin cycle. And minerals such as potassium, calcium, sodium, all of that will be absorbed by plants through roots. That is why we need to add fertilizer to plants, right? Okay. This is one of the questions on the quiz and on your online quiz, where we talked about 9 ATPs and 6 NADPHs are used by Calvin cycle for synthesis of 1 G3P only, isn't it, guys? Yes. So when carbon is fixed and the first stable compound that is formed is made of 3 carbons, and it is called a C3 plants. So whenever carbon is fixed and the stable compound that is formed has 3 carbons only, then it is called C3 plant. What happens when carbon is fixed and the first stable compound that is formed has 4 carbons, and they are called C4 plants. If carbon dioxide is fixed by plants and the first first organic molecule that is formed is actually an acid, then they are called plants. We'll go to that part in a minute. Let's understand why would plants make acids and 4 carbon compounds upon fixing carbon dioxide instead of making C3 compounds. Okay, so what is photorespiration? Photorespiration is a process of using up sugars and not making any more sugars, not ATP, and simply using up all the saved organic molecules in plants. But why would plants do that? It's because plants, when it is dry and hot, in tropical areas and in deserts, uh, plants are undergoing loss of water, which is dehydration. So what they do is they either keep their pores completely closed, stomata completely closed, or partially closed. Because of that, there is less carbon dioxide. When there is less carbon dioxide, what these C3 plants, which are rice, wheat, soy, what they do is they are fixing instead of carbon dioxide, they are using oxygen to add to RUBP. So this rubisco is quite confused in C3 plants because the pores are closed, and the oxygen is not escaping and carbon dioxide is not coming in because the pores are closed, right? Rubisco is adding oxygen to RUBP. When that happens, RUBP is unnecessarily wasted, no energy released, yeah, and no food made. Food is in fact, getting broken down for no reason. So this process is called photorespiration. What is photorespiration? It's a wasteful process wherein rubisco adds oxygen to the Calvin cycle instead of carbon dioxide. But this is not good. It is wasteful because there is no sugar made, there's no ATP made. And plants cannot survive like this. If this is true, then plants wouldn't have uh, survived or evolved so much. But the thing is, some plants have a mutation and they have an alternative. So what they do is they do something smart, and they are called photoadaptations. We'll go there in a minute. But how about rice, wheat, and soy? Yeah, unfortunately, yes, they are producing only 50% of their capability. So, rice crops, they actually, if they can avoid photorespiration by introducing some kind of gene which can stop rubisco from getting confused between carbon dioxide oxygen, then rice crops would make 50% more rice, and same thing with wheat and soy. So yes, whatever you get from rice and wheat and soy crops, non-GMO ones, they're actually 50% of their productivity. They're producing 50% of their produce. Okay, unfortunately that is true. But the GMO ones, I think they are making some they have added some mutation in them, mutant gene in them, and that is making them avoid that process. Well, coming to this, as you can see, photorespiration. This is photorespiration. What is happening in photorespiration is RUBP is taking in oxygen and it is getting broken down, and no ATP is formed, as you can see, right? Well, photorespiration is not a useful process, and the photosynthetic yield is reduced um, so much. It's actually 50%, as I said. So it is draining out the plants. What do plants do? They have some, not all species, but some of the plant species have an alternative method. Luckily, they had a mutation which is helping them to avoid this process. So C4 plants, which are sugarcane and corn, and cam plants, they are doing something else. Cam plants are pineapple and any succulents. So what do C3 plants do? What C3 plants do is they fix carbon dioxide using an enzyme called PEP carboxylase in their mesophyll cells, which is just under the epidermis. The layer of cells is called mesophyll cells. Is mesophyll cells, what they do is they use PEP carboxylase enzyme. And what does PEP carboxylase does is, even when there is little carbon dioxide, because their pores are partially closed, there's very little carbon dioxide, but this PEP carboxylase is not confused. If it sees even little carbon dioxide, it will efficiently capture the carbon dioxide and will quickly synthesize oxaloacetate. And mesophyll cells have enzymes to convert oxaloacetate to malate. Malate through plasmodesmata, which are the channels between the mesophyll cells and the bundle sheath cells, which are the second layer of cells under the mesophyll cells. Malate will pass through the plasmodesmata and is broken down into pyruvate and carbon dioxide. And this carbon dioxide concentration is increased in bundle sheath cells. And rubisco here will capture this carbon dioxide and fix the carbon dioxide or add it to RUBP, and it will start the Calvin cycle. Do you see here, guys, that C4 plants are fixing carbon dioxide twice? First in the mesophyll cells, which is done by PEP carboxylase, which is very efficient in capturing carbon dioxide, and then it is broken down into pyruvate and carbon dioxide in the bundle sheath cells. Again, one more time. PEP carboxylase will capture the carbon dioxide and will make oxaloacetate, which is an organic molecule, which is converted to malate through a chemical reaction. And malate is going to pass through plasmodesmata in bundle sheath cells and is broken down into a pyruvate organic molecule and carbon dioxide. This carbon dioxide, because there's more of carbon dioxide, this process is increasing the carbon dioxide concentration in bundle sheath cells. Now our rubisco is able to add only carbon dioxide to RUBP, and then the Calvin cycle steps will go on. Carbon fixation, reduction, and RUBP regeneration. All those things happen. G3P molecules are formed. Lots of G3P molecules will be made, and they will be made into glucoses or proteins or lipids or DNA or RNA, whatever the plant needs. So C4 plants have PEP carboxylase in their mesophyll cells, which is highly efficient in capturing the carbon dioxide. So these plants do not undergo photorespiration, which is wasting of their RUBP sugar, which is not happening. All the notes are here. Make sure that you pause, read, absorb, and then go to cam plants. What are cam plants? These cam plants are the desert plants. They are determined to save their water because they live in desert. So what they do is they keep their stomata completely closed during all day. But then photosynthesis has to happen when there is light. So what they do is they keep their pores open only during nighttime. During that time, carbon dioxide is going to diffuse through the stomata pores of their surfaces, and the carbon dioxide is fixed by PEP, which is PEP carboxylase, and they make acid organic molecule, and that acid organic molecule is broken down in the morning into carbon dioxide and organic molecule, and that carbon dioxide will be used during Calvin cycle. Again, guys, carbon dioxide is fixed twice. Once, or the first time, during the nighttime, making acids. Acid is broken down into organic molecule and carbon dioxide, and this carbon dioxide is again fixed by rubisco by adding it to RUBP, and then making G3Ps and making sugars. So Calvin cycle right reactions happen during daytime only, but the pores are closed during daytime. So carbon dioxide is collected by keeping the pores open during nighttime. What does this do? This will avoid photorespiration as well as dehydration. So yes, photorespiration is avoided in CAM plants. Guys, remember that Calvin cycle is must, whether it is C4 plants or CAM plants, right? So what is the difference between C4 and CAM? The difference between C4 plants and CAM plants is that they carry out carbon fixation twice. In C4 plants, there is a spatial difference. There is a physical separation. First time carbon fixation occurs in mesophyll cells, and second time carbon fixation occurs in bundle sheath cells. In CAM plants, which are desert plants, crustulation acid metabolism plants, the carbon fixation is occurring twice, once during nighttime, other one is a second time during daytime. So they both have Calvin cycle. C3 plants also have Calvin cycle. The only difference between C3 and C4 CAM is that C3 plants undergo photorespiration, so they have only 50% yield, whereas C4 and CAM plants do not have photorespiration. They have found a way to get away with that wasting of their RUBP. The difference between C4 and CAM is that the carbon fixation is physically separated in C4 plants, whereas in CAM plants it is temporally separated. Guys, temporal means time. Time-wise it is separated. So know all these things for photosynthesis. Pause and read. Make sure you know everything in this slide, all the inputs, outputs. This is a very good picture representation of what is happening during photosynthesis. G3P is a sugar that is made. Two G3Ps will make one glucose. Many glucoses polymerized will make sucrose. G3Ps are converted to amino acids, fatty acids, and everything that plant needs. Know the differences and similarities between chloroplast and mitochondria and photosynthesis and respiration. Guys, there's a difference. Chloroplast and mitochondria are the organelles. Photosynthesis and respiration are the processes. Please know each and every difference and understand the concept. Understanding is important. You have a greatest source in your hands, that is this video. So this video, please make use of this video and use your time well. Good luck. I wish you all the best.